A neuroscientist stands in a sterile, silent lab, holding a vial just removed from cryogenic storage. Inside is a sliver of human hippocampus, the seahorse-shaped region known to house our memories. But he isn't searching for synapses or neurons today. He's looking deeper, smaller, not for the spark of electric signals, but for the silent architecture of memory itself, its molecular fingerprint. Because what if memory isn't just encoded in circuits or the firing of neurons, but in the chemistry that holds those patterns in place? Not just in the idea of your first kiss, but in the physical location of it, in the scaffold of proteins that shape that moment into permanence. Where exactly is a memory? Not metaphorically, physically. Could joy, grief, or terror be etched into the shape of a molecule? For decades, we've imagined memory as electrical, a process of firing and wiring. But now, emerging research suggests something stranger. Long-term memory may depend on stable biochemical arrangements, phosphorylation patterns, methyl tags on DNA, protein synthesis at synapses that don't just transmit memory, but become it. These chemical modifications can last weeks, years, even decades. They're not thoughts, they're structures, physical, persistent, and subject to change or decay. This reframes the mystery entirely. Memory is not a whisper in the brain. It's a form of molecular architecture, one that can be touched, altered, even erased. And perhaps that means it's also possible to heal. In the 19th century, memory was a mystery too elusive for microscopes. Many believed it to be spiritual, a property of the soul, not the body. It wasn't until Santiago Ramon y Cajal, the father of modern neuroscience, peered into the stained webs of brain tissue and proposed something radical. Neurons, not spirits, carried the mind. He suggested that memory might be the physical strengthening of connections between these nerve cells. This idea laid the groundwork for the neuron doctrine, which culminated in Donald Hebb's famous 1949 principle: cells that fire together, wire together. For decades, this was the dominant model. Memory as an electrical phenomenon, a function of reinforced circuitry, encoded in the pathways between neurons. But a strange, forgotten experiment from the 1960s hinted at something else. Psychologist James McConnell trained flatworms to navigate a maze, then ground them up and fed them to untrained worms. Shockingly, the second group learned faster. The claim that memory could be transferred chemically was ridiculed and discarded as pseudoscience. Yet the seed had been planted. Decades later, that radical idea has returned, not as folklore, but as molecular neuroscience. Today, we know that memories don't just depend on synapses. They rely on long-lasting molecular changes within and around neurons. Epigenetic modifications, chemical tags like methyl groups on DNA, turn genes on or off during memory formation. Proteins like PKMZ stabilize long-term memories through persistent activation. Even emotional experiences leave biochemical traces: stress, joy, fear, all encoded through molecular shifts that shape the brain's identity over time. This has changed everything. In conditions like Alzheimer's, memory loss may begin not with neuron death, but with molecular destabilization. In PTSD, traumatic memories don't fade because they're chemically reinforced, too deeply embedded to be disrupted by time alone. We now know that the very fabric of memory is molecular, and that raises profound questions. Could we someday edit or erase memories using targeted molecular interventions? Could we repair the damaged chemical scaffolds in Alzheimer's? Could we prevent trauma from hardwiring itself into the brain? Or does memory, like all biology, decay over time, leaving our identities slowly unwritten? The most unsettling possibility of all: what if your sense of self, the continuity of who you believe you are, is not just psychological, but chemical? What if your story lives not in words, but in proteins, folded and charged, hidden inside the cells of your brain? In 2012, a team of neuroscientists at UCLA made a discovery that would shake the foundations of memory science. They found that a single molecule, PKMZ, or protein kinase M-zeta, was essential for the maintenance of long-term memories. When they blocked PKMZ in rats that had been trained to perform specific tasks, the animals suddenly forgot what they had learned, not just in part, entirely. Their short-term memory remained intact. Their ability to function otherwise was unchanged, but the long-term memory, the part that had been encoded over time through repetition and reinforcement, had vanished. This wasn't amnesia caused by trauma or neural damage. This was targeted molecular erasure, and it opened a door no one was sure we were ready to walk through. If memory could be stored in molecular form, not just in networks or firing patterns, then it could, in theory, be erased, edited, or overwritten at the same level. Could trauma, encoded deeply in biochemical scaffolds, be chemically dismantled? Could joy, could someone else alter our memories, not with hypnosis or suggestion, but with a syringe? These questions were once confined to speculative fiction. Now they were staring scientists in the face. The discovery collided with another rising theory, memory reconsolidation. Neuroscientists had begun to realize that every time we recall a memory, it becomes temporarily unstable, like wet cement. In those fragile moments, the memory can be updated, distorted, or even erased. Recalling is not just remembering, it's chemically rewriting, which means the very act of remembering can make memory vulnerable. It's not just what we remember that defines us, it's the molecular state of the memory when we remember. The dilemma deepens when we ask, what makes you who you are? Is it your accumulated experiences, the things you've lived, learned, survived? Or is it the molecules, the proteins, the tags, the cellular machinery that store and stabilize those experiences? And if someone can manipulate those molecules, what does that mean for identity, for authenticity, for the integrity of memory itself? As the molecular understanding of memory deepens, so too do the questions and conflicts it brings to light. Neuroscientists are now divided over a fundamental issue. Is memory primarily a function of neural circuitry, the pattern of connections between neurons, or is it chemical, rooted in persistent molecular changes within the neurons themselves? This debate reveals a paradox. How can a memory be so stable that it lasts a lifetime, yet so fragile that a brief molecular disruption can make it vanish? Trauma, in particular, offers a haunting example. PTSD patients often recall distressing memories with extraordinary detail, decades after the event. These memories do not fade, they intensify. Molecular studies have shown that stress hardens certain biochemical pathways in the amygdala, particularly those involved in fear and emotional response. This hardening isn't metaphorical, it's physical. Proteins, enzymes, and gene expression patterns become locked in a high alert state, forming biochemical scars that resist erasure. This leads to another frontier, epigenetic memory. Researchers have found that memory may not only reside in neurons, but in the expression of genes themselves. When we form long-term memories, epigenetic modifications, like methyl groups, are added to DNA. These tags don't change the genetic code, but they influence which genes are activated, shaping how neurons function over time. These chemical signatures can persist for years and even be passed down through generations, suggesting that memory, particularly traumatic memory, might leave a molecular legacy beyond the individual. Meanwhile, memory manipulation is no longer hypothetical. Using optogenetics, scientists have implanted false memories into mice by activating specific neural and molecular pathways with light. Others have erased memories of fear by chemically disrupting the same circuit. DARPA has even invested in research aimed at dampening traumatic memories in soldiers, hoping to reduce PTSD risk through molecular intervention. But these developments come with grave ethical concerns. If we can chemically erase grief, should we? Could joy be removed just as easily? If memory lives in molecules, could those molecules be hacked, edited, sold, or copied? What becomes of identity if the memories that construct it can be altered externally? There are already case studies of individuals who experienced profound changes in personality after targeted disruptions to memory-related molecules, not due to trauma, but to drugs, surgery, or illness. They remembered most things, but not themselves. If your sense of self can be lost molecule by molecule, what does that say about who or what you really are? The more we uncover about memory, the more unstable it seems, not in its strength, but in its ownership. And perhaps, in the future, what we remember may matter less than who controls the remembering. The most staggering realization in the neuroscience of memory is this: memory is not just what you recall, it's what your molecules do in response to life. You don't simply think a memory, you become it. At the deepest biochemical level, long-term memory isn't just preserved in the synaptic architecture between neurons, but in the enduring molecular states within them. These are sustained by transcription factors that alter which genes are active, kinase enzymes that keep key proteins activated, and chemical tags that stabilize gene expression for decades. The implications are profound. Memory is biology, and biology remembers. One striking study revealed that two genetically identical mice, raised in different environments, developed radically different brain chemistries. Their neural wiring was nearly identical, but their experiences, the light, the sound, the stress, the play, had rewritten their molecular identity. Their memories weren't just stored in their brains had become their memories. This isn't just metaphor. Experience alters the chemical fabric of who we are, and that reframes everything. You are not a fixed self with a static brain. You are a constantly shifting mosaic of molecular responses, shaped, altered, hardened, or softened by what has happened to you. Forgetting, then, isn't always erasure. Sometimes it's transformation, the reorganization of molecular signals into a new state, a different form of remembering. And remembering isn't just mental recall, it's molecular persistence, the physical endurance of your past, echoing through the structure of your biology. In the end, the twist is not that memory can be altered, it's that memory is alteration. Every experience leaves its trace, not just in thought, but in matter, and matter, in turn, shapes who we continue to become. What science now reveals is that memory is not housed in any single part of the brain, but in a partnership between the architecture of synaptic connections and the biochemical stability that sustains them. Long-term memory requires not just firing neurons, but persistent molecular signals. Molecules like CREB, K-AMP response element binding protein, help activate genes needed to cement memories. BDNF, brain-derived neurotrophic factor, supports the survival and growth of memory-forming neurons. And PKMZ, once thought non-essential, now stands at the center of a crucial insight, that memory can be maintained and even erased through targeted molecular control. This dual model, structural and chemical, has opened new possibilities. In trauma therapy, the concept of memory reconsolidation allows clinicians to guide patients through the recall of painful experiences while subtly introducing new, safer associations. This isn't psychological trickery, it's molecular intervention, disrupting or rechanneling the chemical pathways that keep memories emotionally raw. Already, therapies are targeting these pathways to treat PTSD, addiction, and anxiety, not by suppressing memory, but by changing how the body remembers. The real-world implications are immense. In Alzheimer's disease, scientists now suspect that memory loss doesn't begin with neuron death, but with molecular dysfunction, tiny errors in protein folding, gene regulation, and cellular signaling that destabilize memory before the structure collapses. This understanding has driven a new wave of drug development aimed not at resurrecting dead neurons, but at stabilizing the molecules that keep memory alive. Experimental treatments are now being tested that aim to prevent traumatic memory formation in emergency responders or to strengthen positive memories in those struggling with depression. In the future, molecular interventions might not just recover memories lost to disease, but shield the brain from encoding certain types of suffering too deeply. And yet, the most important shift is conceptual. We are not just thinking minds navigating experience. We are living stories, chemical narratives, written molecule by molecule across a lifetime. Every joy, every fear, every quiet moment you barely noticed, it lingers, not just in your mind, but in your cells. You are not just shaped by your memories, you are made of them. An old woman sits quietly by the window, a worn photograph trembling in her hands. The edges are soft, the faces faded. She stares at them, a family frozen in time, but their names no longer come. Alzheimer's has taken that. The details are scattered like leaves in wind. And yet, something stirs. Her brow softens, a breath catches. There is no name for what she feels, but it's there, a warmth, a quiet recognition that rises not from thought, but from somewhere deeper. Her daughter watches in silence, understanding now what science has only recently begun to reveal. Even when memory seems gone, something remains, a residue, a chemical ghost, a molecular whisper still pulsing beneath the surface. Perhaps memory is not the story itself, not the facts or dates or names, but the way the body still reacts to a voice, a song, a photograph. Maybe the past isn't stored in language, but in the shape of proteins, in the charges of ions, in the molecular patterns that once carried joy, fear, and love. In this light, memory isn't just something you have, it's something you are. And maybe, just maybe, every memory you carry is more than a trace of your life. Maybe it's a molecule that remembers you back.